Hi guys, my name is Saria Chilakuri and I did my presentation on the effects cannabinoids have on the brain and body. Here is a brief overview of some of the main topics I will be covering. Some relevant information to remember is that cannabinoids are a group of substances found in the cannabis plant, and the most common ones are THC and CBD. To go more into detail, we will be focusing on THC and how it alters the brain structure and functions of frequent THC-using adolescents. In Module 2, we learned about various substance abuse disorders and how repetitive exposure can alter the brain structures and functions. I decided to do my presentation on this topic because I found the module on substance abuse disorders to be really interesting because of how drastically drug use can alter the structure and functions of the brain. Here are my learning objectives. By the end of this presentation, I hope to provide you guys with information on how THC works in the brain and how its repeated exposure can lead to various brain abnormalities. The learning objectives for this topic are basically to explain how cannabinoids work in the central nervous system and to describe its effect on brain structure and function. THC in the central nervous system. When marijuana is smoked, THC and other chemicals in the plant pass from the lungs into the bloodstream, which rapidly carries them throughout the body to the brain. The person begins to experience the effects almost immediately. But let's take a closer look as to how these effects occur. Anandamide, a brain chemical, is an endogenous cannabinoid and shares a similar chemical structure to THC. Because of this similarity in structure, the brain allows the body to recognize THC and alter normal brain communication. Endogenous cannabinoids, such as anandamide, function as neurotransmitters because they send chemical messages between neurons throughout the nervous system. They affect brain areas that influence pleasure, memory, thinking, concentration, movement, coordination, and sensory and time perception. Because of this similarity, THC is able to attach to molecules called cannabinoid receptors on neurons in these brain areas and activate them, disrupting various mental and physical functions and causing various effects. The neurocommunication network that uses these cannabinoid neurotransmitters, known as the endocannabinoid system, plays a critical role in the nervous system's normal functioning, so interfering with it can have profound effects. The body's most studied cannabinoid receptors are the cannabinoid 1 and cannabinoid 2 receptors, also known as CB1 and CB2. CB1 receptors are largely found in the central nervous system where they regulate a wide variety of brain functions. CB1 receptors can also be thought of as THC receptors as they are the receptor target for THC. CB1 receptors span the membrane or wall of a cell with its active binding side facing outward. CB1 receptors are like a lock waiting for its key, if that makes any sense. Both endogenous and phytocannabinoids find active CB1 receptors and unlock them. G proteins, located on the inside of the cell, bind to the tail of a CB1 receptor and are released to deliver messages when the CB1 receptor is activated by an agonist molecule like THC. And here are cognitive consequences of smoking marijuana as an adolescent. So in the peer-reviewed research article titled Functional Consequences of Marijuana Use in Adolescents, the authors found that adolescent regular marijuana users performed worse on tests of attention, nonverbal memory, and learning compared to non-using adolescents. Additionally, more days of cannabis use in the past month predicted poor performance on executive functioning and working memory tests, even after years of school completed and verbal intelligence, which honestly kind of surprised me when I learned about this. Early onset marijuana use may help explain some subtle persisting differences in neurocognitive performance over time. Those who start using at an earlier age may have greater susceptibility to the longer-term consequences of cannabis toxicity on the developing brain than those with a later onset of cannabis exposure. And here are the effects of marijuana on brain structure. So users with smaller white matter volumes demonstrated an increased likelihood of depressive symptoms. This suggests that chronic marijuana use during adolescence may influence and affect by disrupting mood-regulating frontal limbic basal ganglia pathways. In fact, brain regions that are rich in cannabinoid receptors and therefore susceptible to effects of cannabis include the frontal cortex, hippocampus, basal ganglia, cerebellum, amygdala, and striatum. Adolescent onset marijuana use has been linked to several brain structure abnormalities, including reduced gray matter volume and density, increased white matter volume and density in non-depressed users, reduced white matter integrity in some areas, larger hippocampal volume but not linked appropriate to learning performance, prefrontal cortex volumes that are smaller than expected in female users but larger in male users, and increased cerebral blood flow in male users. And here are my references, and that concludes my presentation. Thank you so much for listening.